Two-way analysis of variance. For two-way analysis of variance, we need one dependent variable, which is quantitative or scaled in nature. We need two independent variables, which are factors or the nominal variable. They have two or more levels. Two or more levels means suppose they may have the two levels or they may have the more than two levels. In this analysis, we are going to discuss and analyze a complete case study of two-way ANOVA. The data file which is used in this analysis is depression.sav. It is the SPSS format data file. It contains the following variable: the ID of the participants, the gender, the male, female is the nominal with the two levels, male, female. And the next one is birth date, which is not needed for this analysis. The next variable is the BDI, the back depression inventory. This is the scale variable on which we are interested. So this is our dependent variable, scale variable, the dependent variable. So we have to analyze the depression level for the participant, and we want to analyze with the BDI groups. BDI groups diagnosis based on the depression inventory score. And another thing is medicine administered, which medicine is used to control the depression inventory. The two variables are used here. One is BDI, the medicine. This is the factor variable, which is used to analyze this two-way ANOVA. Second thing is the gender, male, female. Along with these two variable, the one more variable is required. That is a scaled variable. That is a dependent variable is BDI. So BDI is our scaled variable, gender, and medicine. These are the two nominal variable. Medicine have more than two factors or the levels, and gender has two levels, male and female. The research question. Our main research question is, did the different medicines result in the different mean BDI scores? It means the average BDI score for the different medicine is same or not. This is the first null hypothesis, along with a secondary null hypothesis, and that is BDI scores are related to the gender in any way. Means the average score of the BDI is same for the male and female, or it is significant. So these are the two null hypotheses that we have to check in two-way ANOVA. The first null hypothesis is H naught. H naught is average score of BDI for different medicines. Medicine number 1, medicine number 2, medicine number 3, and so on. So is it significant for different medicine or insignificant? The null hypothesis is BDI score is insignificant with respect to the medicine. The second null hypothesis is the BDI score, average BDI score for male and female is same or not. The mean average for male, average for female. In two-way analysis of variance, we have another hypothesis which is always be there, and that is interaction. The two factors are independent. There is no interaction. Two factors are what? Medicines and the genders are the two factors. Two factors are independent. This is the null hypothesis. Two factors are independent. And with the alternate hypothesis that there is a significant interaction effect. So H naught can also be written as the interaction is insignificant and H1 significant interaction. H1 is significant interaction. Significant interaction means what? The gender and the medicines are interacted. Means some medicines are specially designed for a specific gender. So they have a combined effect. So that combined effect is known as interaction. So there is interaction, there is no interaction. The alternate hypothesis is there is interaction, there is no interaction. There is interaction. This is the alternate hypothesis of the interaction. So in two-way analysis of variance, we always have three null hypotheses to be tested. One is the hypothesis related to the average scores for the factor number one, the average scores for these are called the simple effects. And then we have to analyze one more effect that is called interaction. So firstly, out of these three null hypotheses, we have to judge whether the two factors are independent or not. So our decision of analyzing these two hypotheses will be based on the third one. So the third hypothesis is to be tested first, and then based on the result of this hypothesis, whether the interaction is significant or interaction is not significant, we have to further decide what type of one way or two way is to be used for the analyzing the simple effects. So in the research question, only the two hypotheses are shown here. One is different medicine results in the different mean BDI scores. Means medicine significantly affecting on the BDI or not. The null hypothesis is that average BDI will be same for all the 3 or 4 levels of medicines, or all the 4 types of medicines. The secondary hypothesis that the average BDI for male and female are significant or insignificant. So these two hypotheses are shown in this research question, but the third hypothesis is not be here, and that hypothesis is what? That hypothesis is the hypothesis of interaction. Assumptions of ANOVA. Before going to test the three hypotheses, we first have to check the assumption. Independent observations. Independent observation means we have to collect the data from the individuals which are not associated to each other. Means we are not analyzing the family members in my samples. We are analyzing the participants which are independent to each other. This is not the independent of variable. This is basically the independent of observations. This is one of the significant effect, because sometimes depression may affect all members of the family. So that we have not to gather the data in a family, we have to gather the data from the individuals or the participants which are independent, means they have no relationship, so that the level of depression will be affecting due to the same factor in the family. The second important assumption is homogeneity of variances. The variances must be same, or we have equal variances. This is called homogeneity of the variances. So Levene test can be used to test this homogeneity of variance. If the Levene test is insignificant, we can proceed to the ANOVA analysis. Otherwise, if the Levene test is significant, we cannot analyze the ANOVA directly. We can apply certain transformation to analyze the data. The third assumption is a very important assumption. In this assumption, you have to check the normality of the data. If the data is normal, then you can proceed. Otherwise, you cannot proceed. Homogeneity can be checked by Levene test. Normality can be checked by histogram or by Kolmogorov or Shapiro test. So I have implemented the histogram here, and that histogram can be made for the frequencies, BDI format, notable histogram. Histogram is constructed and it is more looking like as approximately normal. So the depression inventory is approximately normally distributed. We can proceed for the ANOVA. This is the important thing. The complete flow chart of two-way ANOVA is very important. All the procedures, all the steps of two-way ANOVA is described here. Run the two-way ANOVA in SPSS, whether the sample sizes are equal or not. If the sample sizes are equal, so we have to move into this direction. If the sample sizes are not equal, we can apply the Levene test. Levene test is basically the testing the significance of the variances, whether the variances are same or not. If we have equal sample sizes, the interaction effect is significant. So the very first hypothesis that we have to check into the ANOVA output is the interaction effect. If interaction is significant, then go to this direction. If interaction is insignificant, analyze the data by the two-way analysis. So if interaction is significant, run ANOVA with simple effect. Simple effect means what? Simple effect means what? You have to analyze the BDI separately for male and female. So you have to break the entire data file with respect to the male and female along with the medicines. So what effect there are on the medicine on the BDI for male, what effect there is of medicines on the female. So these two analyses have to be done separately if we have a significant interaction. If the significant interaction is not significant, so we can directly analyze the output of two-way ANOVA. So two-way ANOVA is meaningful to compare the main effects. Main effects are what? Effect of different medicines on the depression, and the effect of uh, gender on the depression. So these are the two main effects. So we have to analyze the two main effect by the output of two-way ANOVA if we have insignificant interaction. If we have a significant interaction, then you have to run the one-way ANOVA 2 times after splitting the file with respect to the gender. Because we have only the 2 levels in gender, so we can split the file for male separately and for female separately, then run a one-way ANOVA for male and then apply the one-way ANOVA for female. If the Levine test is significant, means the variances are not same. If the variances are not same, we cannot apply the ANOVA process. If the Levine test is significant, we can apply Welch statistics. This is nothing. This is the non-pooled t-test. It is a non-pooled t-test that can be used to compare if the variances are not same. We can also have a perception of applying some transformation if the variances are not same. So you can apply a few transformation like logarithmic transformation, reciprocal transformation, square root transformation to make the variances equal, and then you can run the ANOVA directly. So there are the two versions. One, some statistician says apply the Welch statistics. Otherwise, if you can apply the transformation to stabilize the variances which are significant, so again you can move to the interaction effect here. So if interaction is insignificant, directly analyze the output of two-way ANOVA, and then if the two-way ANOVA is significant, means the main effect are significant, so you have to analyze the post-hoc for the both. For the both genders, it has to be analyzed for the both of the different medicines. If the first main effect is significant, like different medicines have a different effect on the depression, so we have to analyze it by post hoc. Means which medicines are giving us a better result, which medicine is not giving us a better result. So that is a post hoc analysis for medicine, and then we can apply the post hoc analysis for male and female. There is I think there is no need to apply the post hoc analysis for male and female, because it has only the 2 levels. Post-hoc analysis is basically applied whenever we have a more than 2 levels. So if after the post-hoc, we have to clarify whether which medicine are significant and which are not significant, that is the end of the process of analysis of variance. Now we are going to implement this complete analysis into the SPSS mean size. Inspecting the mean and the sample sizes. This is just basically we are analyzing the sample means. So these are the descriptive measures. Simple descriptive measures are run by using this code. You can run this code by simply placing cut and copy of this code into syntax, new syntax file, or you can directly run it by menu items. So results are what? Results are the mean for these are the different types of medicines administered. None means no medicine was given to the participants. Placebo means some neutral tablets are given to them. Homeopathic medicines are given to them. Pharmaceutics means some allopathic medicines are given to the participants. These are the different sample sizes. Total 46 sample sizes we have. Then sample means of BDI. These are the means of BDI for males. average of BDI in samples for male, average of BDI for samples in female. Total 54 female participants we have in the sample and 46 male participants we have in the sample. Running the two-way ANOVA into the SPSS, so we have these variables: ID, B date, birth date, or BDI group. So BDI is selected as a dependent variable because it is our scaled variable. Gender and medicines are selected as factor variable. You have to run the procedure by analyze GLM, generalized linear model, univariate. Univariate means we have one dependent variable, which is BDI here. Then you have to click upon the plots. If when you click upon the plots, different files are open, different dialog boxes are open. If you click on the post-hoc to identify the which post-hoc procedure you want to prefer, and you can have options for selecting the how to handle the missing cases. So these are the different screenshots to run the entire thing into the SPSS. The code is given here. If you are not able to run the manuals, just copy and paste this code into the SPSS syntax file and run it. So you will get the same output. These are the outputs of ANOVA. You first have to check the output of interaction. This is the output of interaction. It is one of the very important thing. So initially in the two-way ANOVA, your output is a very important output. This is the output of interaction. Interaction is generally represented as gender times medicine. It is the effect of gender and medicine. Is the gender and medicines are significantly affecting to each other or not? So this is basically interaction, and the p-value is 0.02. 0.02 is an indication that we have a significant interaction. So our null hypothesis of significant our null hypothesis of insignificant interaction is rejected, and our conclusion is there is a significant interaction among the gender and the medicines. Means gender medicines are differently behaving for the males and the females in the, that is the meaning of significant interaction. Medicines are behaving to control the depression differently for the males and for the females. So we cannot analyze the main effects. The main effects are what? These are the main effects. P-values of the main effect cannot be directly analyzed by this output because there is a significant interaction. So if this interaction is not significant, you can trust on these two results. If this interaction is significant, you cannot trust on these two results. So I am ignoring these two effects. These are called the main effect. So I'm ignoring the main effect because there is a significant interaction. So how to control this or how to manage this? This can be done by using one-way ANOVA. So you have to break the files into the two segments. Interaction effect is represented by gender, time, medicine. Means the two qualitative variables are to be here with the product sign is an interaction effect and significant interaction we have. The flowchart says we should now return to the ANOVA with the simple effect. Simple effect for now, ignoring the main effect. Why we are ignoring the main effect? Because interaction is significant. So we have to break our data file with respect to male and female and then analyze the one-way ANOVA two times. One time for males, effect of different medicines on male, effect of different medicines on female. This plot is called a profile plot. Profile plot indicate that one factor which is represented for female, other factors are represented by blue color. So they are interacting. If they are not interacting means they are, if these two graphs are not crossing to each other, meaning what? Meaning that there is no interaction. Suppose our graph of the red marker will be like this. So these are two the parallel graph. One is this blue and one is the dark blue. That is an indication that if we have these two graphs, that is an indication that there is an insignificant interaction. But in my case, red and this blue are interacting, means they are crossing. If they are crossing, it is the meaning that they have some interaction. If they have some interaction, so you have to proceed in the by analyzing the separate data files. You can run this code. This code is sort cases by gender and then split file separated by gender. So these are the two steps that you can do for separating the data file for male and for female. So these are the two steps which are very important. After that, your file will be split into two segments. One is for male, other is for female. Then you can apply the one-way ANOVA with simple effect. So analyze, general linear model, univariate, or you can apply a one-way ANOVA. Instead of going to the generalized linear model, you can go compare means one-way ANOVA. That is another procedure you can follow. These are the steps showing you what to do. BDI and medicine. Only one variable is selected. So second variable of gender is eliminated from here. So BDI for medicine, and it is for only one participant, like it is for male or it is for female. So once you have to select it for males, and then again you have to select it for female. So these are the two one-way analysis. The univariate ANOVA BDI by medicines method is equal to this: include, intercept include, medicine, post-hoc. Post-hoc is Tucky. I already explained you, if you want to analyze which effects are having a higher effect, which are having the smaller effect, so you can apply the post-hoc procedures. And this is the output. So here we have this output for the main effects. This is the result for females only. So you can see this is the result of female only. So means we have the two results. Once we have the result of ANOVA for females, then, and you can see that there is no interaction because we have only one factor variable, which is medicine here. So now we can analyze this medicine. 0.019. 0.019 is smaller. 0.019 is smaller than 0.05, means we have a significant effect of medicine on controlling depression. The significant effect of different medicines we have on the depression. So F statistics is 3.68 and the p-value is 0.019, is a significant interaction effect. The null hypothesis H naught for females, average depression for medicine type 1, which is no medicine, and the medicine 2, average effect on by medicine 2, which is placebo, and effect by homeopathic medicine on depression, effect on by the medicine provided by the doctors, which is type 4. So this hypothesis for females, I am testing this hypothesis only for females. On females, we have rejected this null hypothesis, the significant. So we can apply the post hoc for comparing the effect of different medicine on the different medicine on the depression. Eta square is another thing. Eta square is 0.2. Eta square is 0.208, which is very small. So it's higher eta square is indicating that we have rejected the hypothesis with the high score. A smaller the eta square is indicating that we have rejected the null hypothesis by a very partial score. Means this p-value is not too small. This p-value is not too small, means the partial eta square is a very nominal value we have. So this is an this is the case of eta square is indication that how strongly we are rejecting this null hypothesis. The higher the value of eta square is indicating that we have rejected the hypothesis is by the strong assumption. Smaller the eta square is indicating that we have rejected this null hypothesis by mild variation or by the mild effect. Now H1 is true. H1 is what? H1 is not all medicines, not all medicines have equal effect on depression. Means some medicines have a higher effect of controlling the depression. Some have an insignificant effect on controlling the depression. That can be analyzed by the post-hoc procedure. The next output is the post-hoc. In the post-hoc analysis, the first when the medicines are not given, none is compared with the placebo, and then none is compared with the homeopathic, none is compared with the pharmaceutical. These are the three comparison of the two means. So the null hypothesis is insignificant, means placebo and none, they have a same average effect. There is no effect of placebo and none. Then we have a 0.5, null hypothesis is true, and hypothesis of no effect of homeopathic, and it is same as if we are using the homeopathic medicine and if we are not using any medicine, the both have a same effect. It is not significant. But the third one is significant, 0.021. It is smaller than 0.05. So we can say that pharmaceutical medicines are giving a significant effect on controlling the depression. So homeopathic medicine have no significant effect on controlling the depression. Placebo has no effect on controlling the medicine and the depression, but the pharmaceutical medicines have a significant effect on controlling the medicine. It is for females. Now you have to run it by for the males, and then you have to analyze the output separately for the males. That's all for this analysis.